Hello, everybody. So for today's lecture, we are going to focus on biopsychological research. And we're going to start with this term scientific inference. Okay? Or using the empirical method, biopsychologists measure what they can observe and use these measures as a basis for inferring what they can't observe. So let me give you an example. An individual is presented or presents themselves with anxiety. Okay, the biopsychologist might not be able to see how this anxiety is being manifested. They might not be able to see that person's anxiety just from looking at them. Okay. However, if they hook them up to, let's say, an electroencephalograph and they look at brainwave activity, then they might be able to see, yeah, this is a brain that is not at rest. This is a brain that is very active. This is a brain that is anxious. Okay. So you see how biopsychologists would measure the brainwave activity because that's what they can observe. And then through this, they can infer what is going on in that individual, which is the anxiety. Okay. And we will be exploring scientific inference using human and non-human subjects, experiments and non-experiments, and also through peer and applied research. So if we start off with human and non-human subjects, okay, many questions about the biology of behavior are addressed using human subjects. However, much can be learned also from studying the brains of other species. Okay? And species differences are often more quantitative than qualitative. And what we mean by this is that we're using an animal subject because the brain is smaller, okay? Because the brain is less complex. That's not to say that the same mechanisms are not at play. So if we're looking at a neurotransmitter, dopamine, serotonin, something like that, okay, in that animal model and the human model, we still see those same neurotransmitters, and we see how neurons communicate. We see the receptor sites for those neurotransmitters. The difference is the quantity, okay, the amount of neurons that we're exploring. So by using a non-human subject, it simplifies the process somewhat. Okay, so why use non-humans? Well, they're simpler brains, is exactly what we were discussing, which make it more likely that the brain behavior interactions will be revealed. There's also the comparative approach, which you can gain insight by making comparisons with other species. And there's fewer ethical restrictions for non-humans than with humans. Right? However, I want to preface this that non-human research also requires extensive ethical oversight. And I provided you the guidelines by the APA for experimentation with non-humans or animal subjects. Okay. Why use humans? Well, humans can follow instructions. They make subjective reports, and they are often cheaper to work with because with humans, you can bring them in and out of your laboratory setting or your classroom, or you can go out to them. You can survey them. They can fill out a survey online, all those kinds of things. When you're working with animal subjects, you have an animal lab set up. It's extremely expensive to just care for all those animals, having the proper care for all the cages and bedding and all that kind of stuff, as well as the veterinarian services. But I also gave you the link there for the ethics code for human subjects. Okay, so let's talk about now experiments. Well, the goal of an experiment is cause and effect. Okay, what you want to see is that something changes due to your manipulation. Okay, something is changed because of what you did, right? So if you place people, different people into, let's say, two groups or two conditions, then that would be called a between subjects design. Okay, and if there are any overlapping traits between those two conditions, then you would want to use a between subjects design. So you wouldn't want it to alter both groups. Okay. However, if you're using the same group of subjects tested under each condition, then you would be using a within subjects design. And in some cases, that's that's the best way of looking at it. If you're looking at some kind of a drug that is is possibly helping an individual over a long period of time. So you can increase the drug dosage, you can decrease the drug dosage, and it's based on that same person. Okay. Also, experiments involve the manipulation of variables. Okay. So what you manipulate in your study is going to be the independent variable. Okay. Or you could say that the difference between the conditions is your independent variable. And then the effect of the independent variable is the dependent variable, or what you count. Okay. So if we take, for example, the same scenario with anxiety, okay, let's say that we are looking at an individual, we're hooking that individual up to an EEG, an electroencephalograph, and looking at brainwave activity. Our dependent variable, what we're counting, is the EEG, okay, the brainwave activity. The independent variable is what we would try to do to help this individual. So let's say that we're giving them a particular drug, and let's say that we give them 10 milligrams in one condition, and then 20 milligrams in another condition, 30 milligrams in another condition. You see how those are 3 levels to our independent variable, because that's what we're manipulating, is dosage of this medication or drug. Okay. And then what we're hoping for is that the only reason why brainwave activity is changing is because of that manipulation of the independent variable. Okay. And so then our last thing here is a confounded variable is a variable that affects the dependent variable because it is not controlled for. And so that's why it was so important when I was mentioning the previous scenario, because if the brainwave activity is being changed by something aside from the drug or a confound, then that really messes up our study. And we cannot then say that the drug, right, caused the effect on the brainwave activity. Okay. Now, there's also non-experiments. Okay. The goal here is to acquire knowledge, because in a lot of these studies, you cannot identify cause and effect. You don't have a lot of the things controlled for. Okay. So we're going to talk about quasi-experimental studies and case studies. Okay. Quasi-experimental studies are those in which you use groups of subjects exposed to conditions in the real world. So like I said, you're not bringing them into a laboratory setting. So you're not going to have all those control mechanisms. Okay? It says these are not real experiments, as potential confounded variables have not been controlled for. And one of those things would be random sampling. Okay? It doesn't mean that you don't use these studies. It doesn't mean that these are lesser studies. Okay? It just depends on the conditions that you're working with. You may only be able to use a quasi-experimental study, and therefore you can still, like we said, acquire knowledge through this. Okay. Another example are case studies, where the focus is placed on a single individual or very few people. Okay. So again, not specifically breaking them up into groups and looking at cause and effect between the independent and dependent variable. But case studies are great for gaining a lot of in-depth information about the people. Because you're using so few people, you can really understand a lot about them. Okay. But then again, you have to be careful about generalizability as well. Okay? Because you are only using a very few people, few number of people, or one individual, then your sample size is very small, and you may not be able to generalize the results to the real world. And then lastly, a look at pure and applied research. So pure research is conducted for the purpose of acquiring knowledge. Okay. And this is the work of a physiological psychologist. They would do this in a laboratory setting. They're still looking at, they might be looking at the effects of specific illicit drugs on parts of the brain, that kind of thing. Okay. Applied research is intended to bring about some direct benefit to humankind. So here, the work of neuropsychologists, remember, these are the individuals that are working in possibly a hospital setting, working with post brain damage individuals, and trying to aid them in their activities of daily living and things like that. So you see how this is more of an applied research. But remember, applied research has its basis in pure research. Okay, so if you are uh, as a neuropsychologist, you're identifying a particular psychoactive ingredient or a psychopharmaceutical to give to your patient, the basis for that psychopharmaceutical came from pure research, right? Came out of a laboratory setting. So they definitely build on one another. And then often research projects have elements of both pure research and applied research. And I think I gave you a pretty good example.